next topic of the week at Mote Marine Laboratories Elizabeth Moore International Center for Coral Reef Research and Restoration is coral reproduction. Thank you everyone for tuning in again. My name is Stephanie, and to go along with this lesson, we will also have a behind-the-scenes video, a genetics activity, and further content on our Google Classroom. All of our virtual content that we've been working on is located right here with this class code. So let's get into materials for today. If you would like to grab a notebook and pencil to take some fun notes on coral reproduction, you're more than welcome to. If you also want to grab your dive buddy, let's get ready to dive in. Let's quickly go over our learning objectives for today. First off, we hope that everyone gains a basic understanding of coral reproduction, as well as understanding the processes of sexual and asexual reproduction in corals. And lastly, further understanding Mote's research on this subject. So, how do corals reproduce? To start, reproduction is the process of creating offspring, which keeps species and our ecosystem alive. Now, let's remember corals are sessile, meaning that they cannot swim around and move. As a result, corals have to utilize other reproductive strategies. This includes sexual and asexual reproduction. So we are going to take a closer look to better understand how exactly corals utilize these processes. We are going to start off with sexual reproduction. Most corals utilize this process and are broadcast spawners. Broadcast spawning occurs when millions of eggs and sperm are released into the water column simultaneously. Ocean currents then aid in dispersing them over a wide geographical range. Eggs and sperm will then join and form coral larvae. The coral larvae are called planulae, and eventually they will settle in a good location and then begin to grow their calcium carbonate skeleton. Broadcast spawners can also be referred to as mass or synchronous spawners. Some corals are also brooders, which occurs when eggs and sperm come together within the polyp, and then the planulae is released into the water and dispersed. Mass spawning events occur different times of the year depending on the species and location. Down here in the Florida Keys, it is around the August full moon, compared to the Great Barrier Reef in Australia, which is later in October or November. Corals spawn based on environmental cues in their surroundings. This includes sea temperature, salinity, currents, lunar cues, time of day, usually after sunset, or chemical signaling. With all of this, scientists are able to predict when they will spawn. Sexual reproduction helps increase genetic diversity. Being that all eggs and sperm of one species is released in synchronicity, this increases the odds of gametes mixing for various genetic combinations. This is an amazing video to visually observe spawning. Similar to corals, barrel sponges release their eggs and sperm in synchronicity. It looks like it's a snowstorm underwater. This type of spawning goes along with the idea of safety in numbers. Large numbers are produced to make up for the many hazards, such as predators in the ocean. This can also aid in colonizing new locations, since the larvae is carried by currents and dispersed in a large geographical region. So now we are going to switch over to asexual reproduction and learn the differences. Processes for this include budding and fragmentation. New coral clonal polyps bud from the parents, and this results in new colonies with the same DNA. Fragmentation is the process and technique that consists of breaking the corals into smaller pieces. This allows corals to grow at a faster rate, which is good news for all. Corals reproduce based off of their size, not their age, so the sooner corals can grow to a size where they can reproduce on their own, the better. So let's remember the big difference here. Sexual reproduction increases genetic diversity, where asexual reproduction is the same DNA. Asexual reproduction can be intentional or unintentional. including storms, human disturbance by boats or anchors, or restoration. Now, restoration is intentional down here at our facility in Summerlin Key, where we heavily utilize our fragmentation process in both our in situ and ex situ nurseries, so on land and out in the water. On site, we currently have about 25,000 corals, and in 10 years of work, we have over 80,000 staghorn fragments in our in situ nursery through this process. Now, corals are functionally extinct. They are having trouble reproducing on their own, which is why scientists are stepping in to help, being that the eggs and sperm float and have to find one another. So with less corals to release these eggs and sperm, the harder it is for them to find one another and form the planulae. So our team, as well as scientists all over the world, are doing our part to assist. Our team is always striving for the most efficient research and restoration processes to aid corals. This is some of our footage from Spawning 2019 here in the Keys at our facility. Our team worked endless hours, day and night for weeks, to monitor spawning in our lab, which is a controlled environment. By harvesting parent corals from our nursery and bringing them on site, we were able to keep them under close eye for spawning to further research and aid in restoration efforts. You will see here gametes being released by one of our corals. So to wrap things up today, we're going to go over some key takeaways. First off, that corals are able to reproduce both sexually and asexually. Sexual reproduction increases genetic diversity, while asexual reproduction results in clones. The processes of reproduction include broadcast, mass spawning, brooding, and fragmentation. Also, spawning event for corals is once a year, depending on the species and location. We hope each and every one of you guys learned some fun and exciting facts on coral reproduction with us today. And coming up later this week, come behind the scenes with us and also check out our genetics activity to go along with this lesson. All of this is located on our Google Classroom. If you have yet to join our Google Classroom, here is the information, and feel free to shoot us an email with any questions. Once again, thank you everyone for joining us today. Give us a follow on our social media at Protect Our Reefs, and we will see you guys next week.